Before we continue with the discussion of nucleic acids, let me remind you that you are disciplined, determined, you are dedicated, you are valued, and that you are going to be successful in this class. We are going to continue with the discussion of replication of the molecule of DNA. And when we look at the molecule of DNA, we can make copies of the molecular DNA when we have many assemblies of different enzymes that will work together to make an identical copy before we can have a cell division. So we will have that this process is of extreme accuracy, that you will have 1 error per 10 billion of phosphodiester bonds being made, and the process is really fast. It's going to take place in less than 1 day. In order for replication, in order to make a copy of DNA, the first thing that we need to look for is to unzip or to separate the double strand of the molecular DNA, and that process is going to take place by the use of some enzymes called helicases. There are different kinds of DNA polymerases, but we're going to stay with the simplest kind for this class, and that is DNA polymerases will be the enzymes in charge of making phosphodiester bonds between the sugar units or nucleic acids. Let's overview what we have learned about the molecular DNA. We said that it's very stable, and we will have what is called the Watson and Crick hydrogen bonds between nitrogen bases, where A is always making 2 hydrogen bonds with T, and G is making always 3 hydrogen bonds with C. So we have the ATGC base pair, so that is bringing stability to the molecule of DNA. And on top of that, we also have what is called the stacking. This is the stacking of the nitrogen bases. It is like if they are perpendicular, let's say, to the phosphodiester bond, and they are like stacking on one another, interacting by van der Waals or London dispersion forces. Is by the hydrophobic attraction of these nitrogen bases that is protecting the information, keeping that information, sending the information from one generation to the next. So the molecular DNA, we're going to say that is semi-conservative, and what that means is that you are keeping intact the backbone. What we see here in red is the backbone that is attached to a nitrogen base in each case. So I have a CAGC that was paired to T was paired to A. Now I have transformed that into 2 different templates, and each template is going to act as if it is the blueprint so that we can bring a brand new nitrogen base. And what we see in green is that I have a TA base pair. Now I will have in my daughter strands the same makeup. So we will have a copy that has exactly the same nitrogen bases sequences. We're going to call that to be the parent DNA molecule and the daughter. So we will have, if we have a molecular DNA, it has to unwind. So we will say, yes, I have a 5' end and a 3' end, and I will have the synthesis of DNA in one side. So here we have from one side that will be continuous, the other side will be discontinuous in small little pieces. We will have 2 small pieces of molecules that are being put together to make 2 identical copies. Now the next thing is the molecular DNA is being replicated, or we are making a copy of it in the nucleus of the cell. The function of DNA is to send the information from the nucleus to the cytosol, or let's say to the ribosome, where we will have RNA molecule that is also a nucleotide doing the synthesis of the protein. But once we have separated this molecular DNA, we will have 2 strands, each one of them serving as a blueprint or as the template. One part that is going to be synthesized. So if we have many base pair in here, we have created what is called a replication fork. And this is happening multiple times. So we will have many replication forks. And once we have opened up those replication forks, we will need to use many different enzymes to get the whole DNA molecule replicated. Before we do any details of replication, the first thing that we need to do is to unwrap the molecule of DNA. We say that the molecule of DNA is really long, it's more than 1 meter long, but it's wrapped about these proteins that we call histones rich in lysine. So we have the ends of lysines with NH3 plus that is stabilizing the phosphate unit. Let's say we have this attached to 2 sugars with a negative charge, so we have a stabilization of the molecular DNA by those lysine amino acids that are in the proteins of histones. So we say yes, from being a very long, we will have the chromosomes as it was discussed in a previous video, and then we will have different parts that are called genes, and in the gene what we have is a sequence that is the code for the synthesis of one protein or for the synthesis of an RNA molecule. DNA is really long, but most of the DNA molecule will not codify for the synthesis of proteins. Only some sections will do. But we say that between 2 and 3% will be genes that will be codifying for some proteins. Now, the first step will be the acetylation of those histones. So instead of having lysine, let's say that this is a lysine amino acid ending from the histone, that will become an amide with an acetyl group here. So this acetylation is going to help to unwrap, to open up, to make the nitrogen bases and let's say the phosphate units more exposed for the replication. So that is the first step. It is the acetylation of histones. The second step will be, since sections are going to be unwound, so you have one section that is unwound, we will need to use gyrases or topoisomerases to relax the overwound, because remember that you are unzipping or relaxing or opening up the whole sections of DNA. So we will need to have this second type of enzymes to unwound the molecule of DNA. The next step is to use the enzymes called helicases, and we already have said that these enzymes are in charge of melting or dissolving the hydrogen bonds between the nitrogen bases. So we will need to use helicases to separate the hydrogen bonds between the nitrogen bases. Now that we have seen that, we're going to discuss what is called the Dna polymerase, and that requires the use of activated nucleotide. That means the nitrogen base must be phosphorylated. So it will be deoxyguanosine triphosphate, deoxy-ATP, deoxy-CTP. That means the sugar unit with the triphosphate in order for the reaction to work. The next thing is the use of a primer, and the primer is a very short strand of the molecule of RNA. Now we have 2 templates, so each one of them, but in order to keep it open, we will need to use some enzymes called single strand binding proteins. So I want to go back and say here we have opened up the molecular DNA by the use of helicases. One strand is going to be synthesized continuously and the second one will be discontinuously. We have names for that: the leading strand continuously and the lagging strand discontinuously. Now let's use this picture. It's better picture to explain what's going on. So we have, after opening up, you start with the 5 prime, then from the 5 prime to the 3 prime, that's going to be the leading strand. All at once is synthesized. And then the second strand is called, the lagging strand is made in small pieces of fragments called Okazaki fragments. We see the use of a primer, and then the enzyme that is in charge of placing this primer is called a primase. We also have helicase that is in charge of melting the hydrogen bonds. And what is the function of the primer? The primer is going to provide the 3' OH, but remember that this is a piece of RNA, so it will be a ribose unit instead of deoxyribose. That will be a ribose unit. Now, what will be the function of the DNA polymerase? DNA polymerase is in charge of making sure that the right nitrogen base is the one. So we will have a pool of the triphosphate nitrogen bases, right? We have the nucleotide triphosphate, activated nucleotides, but it's in charge of measuring that the right one is the one making the base pair, plus is also in charge of making phosphodiester bond in the growing nucleotide, let's say the second part of the ladder. So we have the use of DNA polymerase to ensure that the right nucleotide is making the hydrogen bond, and also to ensure that the phosphodiester bond is made. An error will happen, but we will go through what is called proofreading to remove the wrong nitrogen base. I like this picture a lot better because it brings all of the different components that we will need. So we see that this one will be the leading strand. If you see that is made all continuously, we have that DNA polymerase is wrapping both of the strands, but because one of them is going on the wrong direction, it's not going from the 5' to the 3'. You see this one here growing, so this is 3'. So in order to make it happen, we has to make a loop. So when it makes the loop, it's also being synthesized from the 5' to the 3'. So it will always be made from the 5' to the 3'. Now the next thing is what is happening, what is the formation of the phosphodiester bond. So we see in here this is the growing strand. So you see the sugar unit has the OH. In that case, it's no longer the primer because we see that this one is the deoxyribose. So we will make a phosphodiester bond, two phosphoric acid units together. This is called pyrophosphate will be released. So we have, it is the formation of a brand new bond between this oxygen and this phosphorus, and we're going to release these two phosphate units, making a phosphodiester bond. Let me remind you what is an ester. So we say this is an regular ester, and then we have a phosphoester. Because the phosphoester is happening twice, we're going to call that a phosphodiester bond. It's phosphodiester because you have this double bond and this oxygen, and it's in two different directions. So that is the formation of phosphodiester bond. So this enzyme is in charge of ensuring that we have the proper phosphodiester bond, and also that we have the correct base pairing. So the same enzyme is checking that we have both phosphodiester bond and Waxon and Crick base pairing. Now, after we have made different small fragments, we have to use an enzyme that is called ligase, that is only going to work when we have duplex regions. So once you have, let's say, the double strand is made, but there are some little nicks, the function of this enzyme ligase is to create a phosphodiester bond. But look, it must be a double strand section, it must be a duplex. It's only making one bond, is to create this phosphodiester bond, and for that is going to be assisted by an enzyme that is going to create, temporarily is going to create, instead of being just sugar phosphodiester bond, it will be a diphosphate, and then later on it's going to be broken into the right kind.